So the first thing to do is to go on benchling.com, and then the first thing that you would want to do is basically create a project. So you would open this suitcase and you would go to projects right here, and then you would create a new one. We're going to call this one new gene / CRISPR, and then we would create a project. Okay, here we have a notebook where we can add our notes, or you can go here to inventory. In inventory, we can import the DNA sequence for the gene that we're interested in. So it would be import DNA sequencing. Then we can search external databases. And what we're going to do is we're going to search for the gene SCO1. All right. And here we're going to select our genome by going to human genome 38, and here we have all the transcripts. We're going to click accept the default one, and we're going to import it. Once the importing is completed, it's going to show up here under inventory, and now we have the linear map for this gene right here, and we have the whole sequence right here on the left side. Okay, you can always get rid of the split workspace like this, or you can click it. So what we're interested in is actually creating some CRISPR guides. So we're going to go to the left side here on the plus, and we're going to click create, and we're going to go down to CRISPR, and we're interested in CRISPR guides because we're going to be try to knock out this gene. So, and then we're going to do the same thing again. We're going to add our gene and then select the genome HG38. Now we have here 1, 2, 3, 4, 5 transcripts. Okay, and in order to design the guides, we have to really make sure that the guides that we're gonna select is actually exists in all these transcripts. So we're gonna go to show advanced settings, and now we have all of them here, all these transcripts, and we're gonna select all, and then we're gonna do next. And I'm going to click finish. What this does, if we go back to the linear map, is basically it shows all the different transcripts that exist for this particular gene. So we have 1, 2, 3, 4, 5, and then this is 6. Actually, this is just the reference, so we have 1, 2, 3, 4, 5. All right, so in order to, basically we're going to select an exon that has to be in early on in the sequence of that gene in order to design guides to knock out this gene. So if we select exon 1, we have one transcript that actually we're not going to be able to do any editing in. So just in case that was transcribed in our cell line. So what we're going to do is we're going to pick this one. This seems exon 2 seems to be the most ideal one. And one thing we do is basically we avoid the 5 UTR regions, and we also avoid the 3 prime UTR regions. So we're going to select exon 2, seems to be like the best candidate, and we're going to click here, and basically what this does is it selects this exon right here. What we do right now is basically we go to the right side. We have here this design, CRISPR design icon, and we click on design analyze guides, and we do finish. Now we identified this region as this is our target right here, and we go to create. All right, and this creates all these different guides for us right here, this full list. So we have the on target score, and then after a little while, few seconds, the off target appears, the off target scores, and this is these both of these scores, they go between 0 and 100, and the idea is to find the highest. So if you just hover over this, it will tell you that the score is between 0 and 100, and higher is better. And the same thing for off target. It's based on this paper, and the score goes between 0 and 100, and higher again is better. So we're gonna rank him by on target, and then we're gonna try to find the best guide out of this list that actually we can use in order to knock out this gene, to introduce a stop codon. So we take the top one and we see where it actually hits, and this one is actually intronic. It's not doesn't hit all the transcripts, so we're not going to be interested in that one. Try this one, the same thing. So one thing that it does for you is basically it shows where CRISPR is actually going to cut. This is the cut site, and on top here this is the on target score, and on the right side this is the off target score for this particular guide. So if you click on here, it identifies exactly where the guide, which is about 20 base pairs, and it highlights it here on the right side in the list of all the guides. So based on the on target score and the off-target score that you get here, we actually try to determine which one would be best. And usually we try to pick like maybe 2 or 3 guides, just to make sure that at least one would work and knock out the gene, because based on this score, not all of them will have the same efficiency in knocking out this particular gene. You can click on more than one, or actually click on all of them. It will highlight where they actually go, so you can actually use that. Just you can hover over it, and you can click on it, and again it will highlight it here on the right side. And based on that, you can actually so here it seems like that maybe we would want a guide within this particular region right here. That way it hits all the transcripts. That looks to be a good one. Again, that's another one. So maybe these two would be best for this particular gene. And one thing you can do is basically you highlight it. If you're interested in that gene, you can, or you're gonna try to use it for your design. So you right click, it will show copy, you copy it, and it will give you the sequence. It will also give you the reverse complement. So you just right click here and you copy, and here it shows that the sequence has been copied to your clipboard, and it disappears, this message. That's it. So you have your guide, and now you can order it, and you can start with the ligation process as soon as you have the sequence, and you can integrate it into your CRISPR Cas9 plasmid and carry on with your experiment. That's it. This was a brief summary of how to design the guide on CRISPR guides on benchling.com.